Hello, welcome. So today we are going to go through cell division, of course. So uh, this is another human anatomy and physiology lecture. So let's start. Okay, so we are not going to go in details because here we are just going to talk about a few points on how the types of cell division are and whatnot. So cell division, this is a process by which a cell reproduces itself, and there are two types of cell division. Okay, so we have mitosis and meiosis. So let's now go in details now. So mitosis, so each of us begin life as one cell from a fertilized egg by the time we are born. So it consists of billions of cells. So by the time we are born, we consist of billions of cells produced by the cell by process of mitosis. So in mitosis, one cell with the diploid number of chromosomes, with the usual number which is 46 for human beings, will divide into two identical cells, then each with the same diploid number of chromosomes. So this production of identical cells is necessary for the growth of the organism and for the repair of tissues. Okay, so before mitosis can take place, a cell must have two complete sets of chromosomes because each new cell must have the diploid number. So the process of DNA replication enables each chromosome in the form of chromatin to form a copy of itself. So the time during which this process takes place is called the interphase. That is the time between the mitotic divisions. Okay. Then although the interphase is sometimes referred to as the resting stage, resting here means not dividing rather than inactive. So during the interphase, the cell is quite active, producing a second set of chromosomes and storing energy in ATP. Okay. So what are the stages of mitosis? So the stages of mitosis are: one, you have prophase, okay, metaphase, anaphase, telophase, and cytokinosis. Okay, these are the stages of mitosis. So let's now go in details what happens during each stage of mitosis. So starting with prophase, so here the chromosomes coil up and become visible as short rods. Then each chromosome is really two chromatids, so original, which is the original DNA plus its copy, still attached at a region called the centromere. Okay. Then the nucleus, then the nuclear membrane disappears in this stage, and the centrioles move toward opposite poles of the cell and organize the spindle fibers. That's about what happens at this stage. Okay, then let's continue. So which after they organize spindle fibers, which extend across the equator of the cell. That's now about the prophase. Okay, then metaphase, the pairs of the chromatids line up along the equator of the cells. Then the centromere, of which pair is attached to a spindle fiber. Then the centromere now divides. Okay, that's about the metaphase. Okay, then continuing from the metaphase, we go to anaphase. So what happens in anaphase is that each chromatid is now considered a separate chromosome, and there are two complete and separate sets. Okay, so the spindle fibers contract and pull the chromosomes, then one set toward each pore of the cell. Okay, that's what is going to transpire in anaphase. Okay, then under telophase, the sets of the chromosomes reach the pores of the cell and become distinct as their DNA uncoils to form chromatin. Then a nuclear membrane deforms around each set of chromosomes, that's under telophase. Okay, then under cytokinosis, the cytoplasms divide and a new cell membrane is formed.
Mitosis is a process of cell division that makes possible regeneration of body parts. To elaborate, mitosis is the separation of nuclear chromosomes into two identical daughter nuclei. It is divided into four key phases: prophase, metaphase, anaphase, and telophase. An additional phase called cytokinesis occurs after the complete nuclear division. Before a dividing cell enters mitosis, it undergoes a period of growth and rest. This period is called interphase. In interphase, the genetic material of a cell increases. Prophase. During prophase, the chromatin condenses and becomes short and thick to form chromosomes. Each chromosome has two sister chromatids joined together at the centromere. The nucleolus disappears and the nuclear membrane starts to break down. The centrosomes, each with a pair of centrioles, move apart to the opposite poles, forming a spindle between them. Metaphase. During metaphase, microtubules of the spindle interact with chromosomes. This causes the chromosomes to move and align along the middle of the cell or the equatorial plate. Sister chromatids are held at the centromere, confirming the presence of protein structures called kinetochore at the surface. The kinetochore holds the chromosomes and attaches them to the spindle. Anaphase. During anaphase, the sister chromatids separate at the kinetochores, and the daughter chromosomes, along with the divided centromere, move towards the opposite poles as the microtubule shortens. The poles of the spindle also move apart and help to separate the chromosomes. As the chromosomes separate completely into two sets, one at each pole, anaphase is considered complete. Telophase. During telophase, the daughter chromosomes arrive at the pole and the chromatin starts to decoil. Also, the nuclei reform along with nucleoli. This triggers the reformation of the nuclear membrane. Cytokinesis. Cytokinesis is the partition of the cytoplasm. In animals, a cleavage furrow appears in the plasma membrane, which deepens and divides the cytoplasm into two daughter cells, each containing a nucleus. In plants, new cell wall formation is triggered at the center of the cell by the formation of a cell plate. The cell plate grows outwards till it meets the existing wall, resulting in two daughter cells. In this module, you have learned mitosis is the process of separation of nuclear chromosomes into two identical daughter nuclei. Mitosis involves four key phases: prophase, metaphase, anaphase, and telophase. In prophase, the chromatin condenses and becomes short and thick to form the chromosomes. In metaphase, microtubules of the spindle interact with the chromosomes. In anaphase, the sister chromatids separate out at the centromere and move towards the opposite poles as the microtubule becomes shorter. In telophase, daughter chromosomes arrive at the pole and chromatin starts to decoil, followed by the reformation of nucleoli and nuclei.
Then we are done with mitosis. Now let's go to meiosis. So meiosis is a more complex process of cell division that results in the formation of gametes. Then a gamete is a cell that fuses with another cell during fertilization, such as the egg and the sperm. So cells in meiosis, one cell with the diploid number of chromosomes divides twice to form 4 cells, each with the haploid number, which is half the usual number of chromosomes. So in women, meiosis takes place in the ovaries and is called oogenesis. Then in men, meiosis takes place in the testes and is called spermatogenesis. So the egg and the sperm cells produced by meiosis have the haploid number of chromosomes, which is the 23 for humans. So meiosis is sometimes called reduction division. So because the division process reduces the chromosome number in the egg or sperm. That's one thing you have to know. Then during fertilization, in which the egg unites with the sperm, the 23 chromosomes of the sperm plus the 23 chromosomes of the egg merge and restore the diploid number of 46 in the fertilized egg. So together they make up the 46 chromosomes after they come together, the egg and the sperm. Okay, so that's the proper number of chromosomes. Number are maintained in the cells of the new individual. Okay, so that's the summary about meiosis and mitosis. Okay.
Meiosis 1. In this module you will learn about meiosis 1. Meiosis is a nuclear division that occurs in the germ cells of sexually reproducing organisms during the production of reproductive cells called gametes. Meiosis is a reductional division that reduces the chromosome number of a cell by half. Meiosis involves a single round of DNA replication followed by 2 cell divisions, meiosis 1 and meiosis 2. The entire process of meiosis results in the formation of 4 haploid daughter cells with half the chromosome number as that present in the diploid parent cell. However, after fertilization occurs, the chromosome number gets restored. Meiosis 1 is the reductional division in which the chromosome number gets halved and distributed between 2 daughter cells. Meiosis 1 includes 4 stages: prophase 1, metaphase 1, anaphase 1 and telophase 1. Let's know about the homologs before studying the phases of prophase 1. Each diploid cell has 2 copies of chromosome, one inherited from the male parent and the other from the female parent. These 2 copies, coming from different parents, are similar to each other and are called homologs. Now let us look at the different stages of prophase 1 in detail. To begin with, let us learn about leptoteme. During leptoteme, the chromosomes condense and become visible. The zygotene phase is followed by pachytene. During pachytene, the chromosomal crossing over now takes place between the non-sister chromatids of the homologous chromosomes. This crossing over is what makes each individual unique and causes the recombination modules to be visible. As the crossing over completes, it marks the end of the pachytene phase. Diplotene is followed by diakinesis, which is also the last stage of prophase 1. During diakinesis, the chromosomes are fully condensed, the nucleolus and the nuclear membrane dissolve, and the mitotic spindle begins to assemble. Prophase 1 is followed by metaphase 1. During metaphase 1, the microtubules from the opposite poles attached to the chromosomes at the kinetochores, and the bivalents align along the equatorial plate with a random orientation. Either of the parental homologue takes up any site on the spindle. The metaphase 1 is followed by anaphase 1. During anaphase 1, the homologous chromosomes separate out and move to opposite poles with shortening of the microtubules. The sister chromatids, on the contrary, remain attached at the centromere. In this module, you have learned meiosis is a reductional division which reduces the chromosome number of a cell by half. Meiosis involves a single round of DNA replication followed by two cell divisions, meiosis 1 and meiosis 2. Meiosis 1 includes 4 stages: prophase 1, metaphase 1, anaphase 1 and telophase 1. Prophase 1 is further subdivided into 5 stages: leptotene, zygotene, pachytene, diploidine and diakinesis. During leptotene, the chromosomes condense and become physical. During zygotene, the duplicated chromosomes pair with homologues to form bivalents. During pachytene, the attachment of homologues to each other along their length, that is synapsis, is complete. In diploidine, synapsis springs to break down, the chromatids start to pull apart, and chiasmata are visible. By diakinesis, the chromosomes are fully condensed, the nucleolus and nuclear membrane dissolve, and the mitotic spindle begins to assemble. Prophase 1 is followed by metaphase 1, when the microtubules from opposite poles attach to the chromosomes at the kinetochores, and the bivalents align along the equatorial plate. During anaphase 1, the homologous chromosomes separate out and move to opposite poles with shortening of the microtubules, forming 2 haploid sets. During telophase 1, the chromosomes reach the poles and nuclear envelopes are reformed around the 2 daughter nuclei and the nucleoli reappear. A furrow appears in the cytoplasm and the cell divides into 2 by cytokinesis.
In this module, you will focus on meiosis 2. Each daughter cell formed after meiosis 1 inherits only one of the 2 homologous chromosomes, and each homologue has 2 sister chromatids. Meiosis 2 involves the separation of these sister chromatids to produce haploid cells. Therefore, meiosis 2 is an equational cell division. As both the daughter cells produced from meiosis 1 undergo meiosis 2, the final result of meiosis 2 is 4 haploid daughter cells. Like meiosis 1, meiosis 2 can also be divided into 4 stages. These are prophase 2, metaphase 2, anaphase 2, and telophase 2. Prophase 2 is very short. During this phase, the nucleoenvelope and the nucleoli disappear and the spindle is formed with the help of the microtubules. In addition, the chromosomes recondense during the prophase 2 and become compact. Prophase 2 is followed by metaphase 2, during which the chromosomes align along the equatorial plate and the microtubules attach to the kinetochores of the chromatids. Metaphase 2 is followed by anaphase 2. In anaphase 2, the sister chromatids separate out at the centromere and move towards the opposite poles as the microtubules become shorter. Anaphase 2 is followed by telophase 2, which also marks the end of meiosis 2. During telophase 2, the nucleoli are reformed and the haploid nuclei get enveloped in membranes. In addition, the chromosomes decondense and the spindle disappears. Division of the cytoplasm, cytokinesis, results in 4 haploid daughter cells. Meiosis plays a significant role in the life of sexually reproducing organisms. It results in gamete formation and helps conserve the chromosome number in sexually reproducing organisms. All the 4 gametes produced are genetically unique as they are produced by the crossing over of homologous chromosomes. The genetic uniqueness of the gametes is further attained by the independent assortment that results in each daughter cell receiving either homologue from each chromosome pair. Let's summarize what you have learned. Meiosis is a specialized nuclear division in which the chromosome complement of a cell is halved. Meiosis involves 2 steps, meiosis 1 and meiosis 2. Meiosis 2 has 4 stages, prophase 2, metaphase 2, anaphase 2, and telophase 2. Prophase 2 is very short. The nuclear envelope and the nucleoli disappear and the spindle is formed. The chromosomes recondense and are compact. During metaphase 2, the chromosomes align along the equatorial plate and the microtubules attach to the kinetochores of the chromatids. In anaphase 2, the sister chromatids separate out and move towards the opposite poles. During telophase 2, the haploid nuclei gets enveloped in membranes. The chromosomes decondense and the spindle disappears. Cytokinesis results in 4 haploid daughter cells.
So we are now done. This was more of a short summary, not necessarily going in details. Okay. So thank you for watching. If you haven't subscribed, please subscribe and hit the bell notification. And see you on the next tutorial video. Okay. Thank you.